talking with Dr. Joe Barbieri, and he's a professor of microbiology and molecular genetics. Yep. And today is July 20th, 2016. First of all, Dr. Barbieri, if you could tell me a little bit on how did you wind up having an interest in science and pursuing a career in the biomedical sciences?
As I went through school, I became really interested in really mechanistic science. So I was a biologist, but the things that interested me were genetics and biochemistry. And I was really fortunate in having a microbiologist in my senior year as a undergraduate really turned me on to the biology and the diversity of microorganisms. And I went to graduate school at that point to study genetics. And this was at the University of Massachusetts, Amherst. And it was an interesting situation because I had actually identified the faculty member that I wanted to do my PhD research with, and UMass had a requirement that you had to do 4 laboratory rotations, and I was always trying to get out of the laboratory rotations. But the final laboratory rotation, I worked in a lab that was studying pathogenic spirochetes, and it was my first real experience in studying pathogenesis and disease in the microbial world, and it really turned me on. I was really excited about it, and it was really sad for me to have to go back to Kurt Thorne was the investigator, and tell Dr. Thorne that I was going to study microbial pathogenesis instead of genetics. And it was and I thought, I didn't know how he was going to respond, but he was actually really happy for me, and that I had found my niche, and it was really the first time I realized what academics were all about, and all the mentoring that you get is really built around trying to help you figure out what you want to do, and then the payback is that later on you get to do that also. So I studied the physiology of microorganisms, and what I found is I was more of a reductionist than a systems person, and that pushed me into the field of pathogenesis that was more related to toxin biology than to organismal biology or genetics and physiology. So I went when I finished my PhD, I went out to UCLA. And I worked for an investigator, John Collier, and John had just discovered the first mechanism of action of a bacterial toxin. It's an adiparabosylation reaction, and he was a special individual. And
now, what years would this have been?
Yeah, this was now. I received my PhD in 1980. And I went out to UCLA just right after that. So this is the early 80s now, and in the world of science, we were just getting into the really molecular and recombinant DNA technologies, and they were getting popular with laboratory bacteria like Escherichia coli, E. coli, but not in the pathogens. But Collier was one of those individuals that was really pushing all of these molecular approaches utilizing bacterial pathogens. And it was just a really exciting time. And I began to study protein toxins as a field, and I had a great project studying the heterogeneity in a population of toxins that we would prepare in the laboratory, and it was really a basic research project, and we were able to identify a nucleotide that was bound to a subpopulation of the toxin, and it turned out to be an interesting observation, but later on it actually was required to crystallize the toxin. So that was my first really interaction I had with a crystallographer, David Eisenberg at the Molecular Biology Institute at UCLA. And I began to realize actually how important, if you're going to study toxin biochemistry, how having a structural element was really was really key to that. So and since then I've always interacted with a lot of structural biologists.
And so in the process, I assume you were publishing then some of your first papers then
Right,
too?
right. Yeah, I was really fortunate. The first year I got into the laboratory, we identified this nucleotide, and I actually had a publication in my postdoc after my my first year of research, and that was exciting for me to work with John Collier, because he's a tremendous writer, and I thought I was a pretty good writer, and then he actually sat me down and taught me about scientific writing. You know, he was one of those scientists that he really defined the study of pathogenicity in relationship to toxins. First studying catalysis, then how toxins interact with the host cells, and then the processes of protein translocation. So he drove that field for almost 40 years. And we became friends over time. And in 1984, we moved the lab from UCLA to Harvard Medical School, and that was a great experience for me, because I hadn't been at a freestanding private medical school before that. And all of my training as an undergraduate and at this State University of New York at Cortland, University of Massachusetts Amherst, and now UCLA had been all public institutions, and I didn't really appreciate what a freestanding medical school would be. And it was my first experience, and it was quite the medical school to do it at. Really exciting, but it made me appreciate what an institution is like when they're focusing on biomedical research as a topic. And I was really that was really influential on me as I started looking for faculty positions, and as I looked for a faculty position, I was looking for a medical school that would have a research effort. We were from I was from New York. My wife was from upstate New York, and we sort of drew a line out to the Mississippi River, as far as west as we would want to go. And fortunately, we didn't say Lake Michigan. And as I started looking around, a position opened up at the medical college, and I got my map out, and we checked it all out, and it looked like a really interesting situation, in that you had this city that was just north of Chicago, which had a lot of medical schools, and Madison was west of us, and here was this freestanding medical school, sort of setting in a fairly large city all by itself. And I think that was my only real strategy in looking at that, and having been in Boston, where there are just innumerable numbers of medical schools, to have a medical school as the only medical school in a city being pretty unique. So that was my strategy. And then when I came out to interview, I was really I was taken by the collaborative nature of the faculty, and although we had different departments, there was a lot of collaboration amongst the various departments. People like Kent Wilcox and John Lau and Tim Herman and Vaughn Jackson and Bill Cashdollar. Just a great number of people that were doing research, collaborative research, but outside the departments, because we were either in cell biology or microbiology or biochemistry. So when I interviewed for the first time, I interviewed in a number of different departments, and I met Sid Grossberg, who was the chair of the department, and the thing I remember about that interview is that I think Sid had a grasp on his faculty's research as well as the faculty did, he really struck me as a very bright individual, and he was really at the prime of his research career, having standardized interferon as a numerical unit, and that was a very significant advance for the virology field. And then as I interviewed, and I had a good time, and they invited me back for a second interview, and my wife and we had a daughter at the time. She was about 18 months old, and I remember coming back, and the first person I interviewed with was actually not Sydney, but it was Betty Sue Masters, and Betty Sue was the chairman of biochemistry, and she was actually the first director of the MDPhD program, and that'll turn out to be important a little bit later on in my career. But I went into her office, and she looked me straight in the eye, and she said, "You are coming, aren't you?" And I was like, "Wow, this is a different chair that I." And it gave me a perspective of her, where she was just really proud of the institution, and really more than just the chairperson of biochemistry. She really spoke for the medical college. So with all of that enthusiasm, we came out to Wisconsin, and that was in 1986, and it's now 2016. So I've really enjoyed it here.
Well, now, you came from Harvard. Before that, you'd been at UCLA.
Right.
You were at some of the nation's top research institutions. Mm-hmm. And when you came here in 1986, research was really just getting off the ground. Mm-hmm. And so, was it a culture shock for you?
Well, that's I think it was for a lot of the investigators that were leaving the East Coast. I think one of the I was really fortunate in that I had trained with really a top investigator, and he trained me to be able to make circles, which basically means you can ask a question, you do experiments to solve that, you write about it, and you make another question. And my research was really self-contained in the sense that I could do all of those things, and I learned to do them better as I worked along. So I had a reasonable amount of confidence in my ability to do research. But what I really looked for when I came here were individuals that I could collaborate with. In my second visit, I met a guy named Henry Majorco and Art Haas, and I'm going to say a guy because they were just a lot of fun. So they were a little bit more senior than I was. I think they were associate professors at the time,
and they were both in biochemistry.
They were both in biochemistry, and I probably spent more time up in biochemistry for my first 2 or 3 years here than I did in microbiology, and that's really what toxin researchers do, is that we do collaborate. So when I came back for the second visit, I really made sure that there was a really strong classical biochemistry department that I could collaborate with, and those two individuals really turned out to be not only good collaborators but really great colleagues. So again, it was this collaborative nature of the research that toxin researchers do that really grounded me at MCW. It was a fun time because we were building. And as a new investigator, faculty and chairs and deans were interested in our opinions on where science was going and how to build competitive programs. Where I'm sure if I were out at a more established institution, research-wise, I would have been, on the bench and hanging out, there without any interest from others. So it was really I think it was really a special time to be here, and and I guess the other thing that always struck me that I rarely heard from my colleagues is how supportive the clinical faculty were of research. And the example I'll use is that, so I studied protein toxins, and I was just shifting my research from diphtheria toxin, which John Collier worked on, to pertussis toxin. And I did that because there was a public outcry to develop an acellular pertussis vaccine right about the time I came to MCW. And I remember going down, after I'd gotten my lab set up, Sydney arranged for me to go down and see Starkey Davis, who was the chair of pediatrics, and the hospital was still down by Marquette. And I remember going down there, and anybody that knows Starkey, I didn't know him, but I got to know him really well. Is there was a cowboy hat in the corner, and really sharp polished boots on his,
he was
on
from
his
Texas.
footery. He was just a great guy, and he was interested in what I was doing, and he I don't know if I should say this here, but he actually wrote me a check to help support my laboratory as I got started, and literally after this conversation, and I was just flabbergasted that a chair of a clinical department would first take the time to talk to me about the research I was doing, and then would actually step up and physically support it. I remember going home that after that and telling my wife Cindy. So I was really fortunate in that I was doing molecular pathogenesis when I came to MCW. So I had a lot of recombinant experience in manipulating DNA, and I had a good background in protein chemistry. So the work that I did was really building derivatives of toxins and studying. It was sort of called reverse genetics. Instead of selecting for mutations in the environment or in a test tube, we actually engineered them based upon the informatics, the limited informatics that we had in our field. And I'll just to put that in perspective, when our first database of protein sequence was on a 5.25-inch floppy disk that we would buy from a company, and it had actually 3 protein sequences on it. So your protein either typically looked a lot like albumin when you line things up, but that's to compare to the probably millions of sequences that we have now. So I was really fortunate in that I learned informatics and protein evolution really from the leaders in the field that were really doing families of proteins that had common function, that were related to each other, and we built algorithms to analyze the relationships amongst them. So
now, when did you get your first grant?
That's a fun story. So I came out to I interviewed in I guess I signed my contract in September, and I actually went back to Harvard, and I went back to Harvard to write an R01. So I wrote my R01, and then I must have flown out here to submit it, because I had to be at MCW to submit it, and I submitted it on October 1st, and then I went back to Boston to finish some experiments, and I came back in November. So I started in November, and I was really fortunate in the first grant that I wrote was funded by the NIH.
Wow.
And I've been really fortunate over the years.
Now, which agency within the NIH?
I'm sorry?
Which agency?
Oh, NIAID,
Okay.
Allergy Infectious Disease.
Okay.
So that's been my home institute for for the last, well, for a long time, because I also had a fellowship as a postdoc that was out of NIAID also. So I've been funded by NIAID probably for about 35 years now.
Now, you mentioned with Dr. Collier that he was a good writer.
Yeah.
And as you wrote a grant the first time,
Yeah.
was it hard? Was it a challenge, or did you have the benefit of having worked with Dr. Collier
Yeah.
that you knew the importance of writing?
Yeah, that's really what John taught me, was to write, and he taught me to write in a very intense way, in a very focused way, and actually I found it that really it wasn't that I didn't. It really was the speed at which I wrote. That was my rate limiting step in all of the, and probably up until even now it is, I think. But it really is the concept that you work on a document until you've completed it, and then move into another area. So I'm very linear in the way I write and I read and I work, and I think that's a product of having trained with John. He taught me that, and I think one of the things that really influenced and helped me out early on is that I had a series of MD-PhD students who received their PhD in my laboratory in sync. And the first was Galen Cortina, and Galen was he was just a great kid. He was from UCSB, University of California, Santa Barbara. And I remember he interviewed in the dead of winter out here, without a coat, and I actually went out and got him a coat, and I don't know if that influenced him as being his mentor later on, but I was able, but we did do that, and he worked in my laboratory, and we just had a great time together. I had a technician, Irene Capuano, and Galen and myself, and the three of us worked on the bench, and we did some really nice structure function studies of pertussis toxin.
Well, now, with your work on the pertussis toxin, then you developed a name, and this was your niche, and people knew the work that you were doing. Mm-hmm. You started to develop collaborations with researchers at other institutions and even outside the United States. Right. How did that happen?
Yeah, I had it was really pretty fortunate. The story, the way it goes is as I was leaving, and my first grant was actually on pertussis toxin. I spent the last half of the year purifying the toxin from Bordetella pertussis and characterizing the toxin and developing a genetic system to study it. And I had there was another postdoc in another laboratory at Boston University. His name was Rino Rapoli, and we were working on the protein, and he was doing genetics, and we contacted each other. We were both postdocs, and he was getting ready to go back to Italy, and just as we had developed the first expression system for the toxin catalytic subunit, because we had that gene, Reno had cloned the whole operon, and we developed this collaboration, and it was just a lot of fun because he went back to Siena, and if you're going to collaborate, I think that might be the best city in Europe to collaborate with. And we got to be really good friends. And he actually went back to Siena, his hometown, and he built a company called Sclavo, and they made some of the early pertussis vaccines. So we continued our collaborations over the years in working to develop a genetic pertussis vaccine, and we published together the first recombinant full-length form of pertussis toxin that could be used as a vaccine candidate, and that was I think it was 1989, and that was really a science article. It was actually a big group, a lot of us doing a a lot of research, but really out of three laboratories, and it was a lot. That was a lot of fun.
Well, that's a significant achievement. What's the status of that now? Is that a vaccine that is?
It's actually it's an interesting story in that there are a few. So it didn't. It wasn't pursued as a vaccine because at that time recombinant proteins weren't used as vaccines. So they used a formalin inactivated form of the toxin. But even today, it's very clear that these recombinant proteins are much better at vaccines than the chemically inactivated forms, and that's what we use on some of the toxins that we study now. But it was simply the public not being ready to accept a recombinant protein in a vaccine. But it was important because we were able to take it from an idea actually to a vaccine product. And maybe someday we will see that, but there are some the pertussis vaccine has always been a challenge to eliminate the disease. We've been able to eliminate the pathology, but not actually the disease yet. So there's still room for working on that.
And you also were involved with some Japanese researchers as well, if I recall correctly.
Right. Yeah. So it was three laboratories that were all sort of working on different aspects of the toxin, and each of us made a really nice contribution to that project. And that was Reno was a dear colleague, and it seems like all the way through my time at MCW that I've always had an important colleague to work with, and I think that it just reflects on how we do research in protein biochemistry. You just need a lot of people with different expertise to be able to move forward. And as I am about 2 or 3 years after coming to MCW, we recruited Dara Frank out of Barbieri's lab. And Dara and I have been colleagues and close friends for the last 27 years. And her research, she was a Pseudomonas biologist, and I was studying these protein toxins. And we got together with another of our MD-PhD students, Scott Kulik, and we were able to clone one of the major virulence factors out of Pseudomonas aeruginosa, which is important pathogen for patients that have cystic fibrosis or burns or trach patients. But we were able to clone the gene, and it was the first gene of a very large family of proteins that were involved in what we call type 3 secretion in delivering toxins not out of the bacterium and into the medium and then finding a cell, but actually directly injecting the toxin into the cell. And they're called type 3 cytotoxins. And that actually Dara still works on one of the 4 type 3 cytotoxins that were part of the arsenal that Pseudomonas aeruginosa has. And she's done really, really elegant research on that. And we worked in that field for about 10 years, and I think the best projects that we did in there was that we were able to understand how one of these toxins was able to inactivate RAS, which is a very important signaling molecule inside of a cell involved in regulating proliferation, and it did it through a really unique mechanism that one of my other MD-PhD students, Anna Ganesan, was able to work through. And Anand is he's really exceptional. He's now an associate professor at UC Irvine. He's a dermatologist by clinical training, and he's a biochemist by research training, and very successful, very well funded by the NIH. And I see Anand as being a leader in biomedical research as we move forward. We just had him back maybe 2 or 3 years ago to give the keynote at our MDPhD graduation dinner, and it was just exceptional.
Now, during the years then that you've been at MCW, during Dr. Cooper's tenure as dean, and then when Dr. Dunn became dean, research really started to pick up, and we really emerged as one of is a major research institution.
Yeah.
And so it really was a change. You saw the Mac Fund building going up, the TBRC, the HRC building. So you saw expansion of the number of labs here and all. How did that impact the institution? How did that impact your research? Were there new colleagues for you to work with as we expanded as an institution?
Yeah, I think that was, we really what happened was when I first came here, there were a handful of investigators that were collaborating within each of the among the various basic science departments, and then and as the school grew, that theme remained, but we began to collaborate with different subsets of individuals. So the collaborative nature of MCW is really at its roots as far as research goes, and it just continues to grow, and it's grown through these centers that have been developed, as well as in the basic science department. So I think that's something that when people come to visit, my colleagues come to visit MCW, they really do recognize that, they meet a lot of people that are doing research that are really excited about what they do. But what they always comment on is that it's amazing how many people were really interested in the research that they were doing. And I think that's part of the collaborativeness of MCW, is that we really do like to hear about research as a group.
And you mentioned, the all of a sudden the development of centers, the cancer center, cardiovascular center, but also basics. What you saw were clinical departments had basic scientists that were also on their faculty and were housed within the clinical department. So it really was a significant change with sort of the engagement of basic sciences really in the clinical departments as well. Yeah. Did you continue to work with pediatrics?
I did. I've always had an interest with the pediatric faculty, and I was fortunate in that I was able to bring a couple different pediatric fellows into the laboratory, and I guess the fun part about that was that they always had to do a research paper, and it was really sometimes a challenge for us to, in a year or so, to get enough of research that a pediatric fellow could do to be able to write a paper. But Mark Splingard was a good colleague in pediatrics, and a physician, and he was always very supportive. And Mark was always able to help us acquire enough of patient samples to be able to make a research project for our various fellows. So it was I always had a really great relationship with the pulmonary group in pediatrics, and we continue to do that. Dara does it a little bit more than we do now. We've done some transitioning as I finished with my fourth MD-PhD student in series, Matt Reese, and Matt was a math major from Madison, who came and joined our MD-PhD program, and he came and worked in my laboratory, and he studied some of the RAS signaling that this Pseudomonas toxin catalyzed, and Matt was just a spectacular scientist, and he went to the Brigham to do his residency, and then to Penn to do his fellowship, and then came back to MCW, and he's a faculty now at the Blood Research Institute, and he does his clinical interest is in prostate cancer, and he's just been great to see him develop as a student and a scientist, and now a colleague as a faculty member.
Well, now we've talked a lot about your work as faculty member in research, but you've also been a teacher all these years as well.
Yep.
And as you've taught, as you've talked with the medical students, but then also with the graduate students, how do you approach teaching? How do you inspire, encourage, especially our graduate students?
Yeah, I think that what the whole goal of biomedical research is to make students independent, to make them independent investigators, and you have to work with a student, and you have to introduce concepts to them, but you have to also give them the opportunity to think about problems and make hypotheses and test them, and to work with them in experimental design, and help them develop as free thinkers. I guess that would be the way to think about it, because ultimately their success is not going to be based upon the actual research that they did in your laboratory, but did they learn enough to be an independent investigator, to be able to drive a research project on their own, and knowing that you'll never be there to drive them. And I've been really fortunate in that I've had a number of PhDs that have done really quite well. Anthony Moresso was part of the Pseudomonas team, and he went down to the University of Chicago and worked with Olaf Schneewen, and now he just got tenure at Baylor School of Medicine, doing really well, and we talk once in a while, and he'll call me up, and if he has a question, or I'll call him up if I find that I found something that he's done really well. So we do have a lot of good interactions, and I think my best student is Kathy Krieger, and Kathy is at Abbott Laboratory. She's a neuroscientist, and best being, just having the heart and soul of a basic scientist. And she went to work with Bob Lefkowitz at Duke, and then went to Abbott to come back to the area. And it was interesting. She could have been an academic scientist, but she chose the research in a large pharmaceutical company to be a little bit more attractive to the way she wanted to do her research. So she's done very well, and a great role model for women in science.
Now, how did you wind up taking on the MD-PhD program, the Medical Scientist Training Program?
Sure. So I was really fortunate in up to as I said, I had 4 MD-PhD trainees over a 12-year period, all very successful students. They completed their PhDs in 3 years each, all of them, published very well, went on to do great residencies, and they're all very accomplished scientists now. So right after that, there was a transition in the MD-PhD program. Sid Grossberg had been the chair for about Sid he was the chair of micro, but he was the director of the MD-PhD program for about 11 years.
I forgot that.
And as we and there was a transition in Dave Waltier was asked to be the director. Sid stepped down his directorship, and Dave was a card-carrying physician scientist. He was the associate chair of anesthesiology, very, very accomplished. John Campine was the chair of anesthesiology at the time, and John was a really, really strong proponent of MD-PhD training and research. Him being a physician scientist, also an MD-PhD. So when Dave took the program over in 2000, he asked me to be the associate director, and I think the reason for that was that I had a lot of experience in the basic science departments, and that was a part of the program that I could contribute to, helping students find a research laboratory to work in. And so I think we had a great relationship. The program went from having 2 students per year enter the program to having 4 students per year, and it gave us a little bit larger critical mass.
And the competition is pretty fierce.
Oh, it is. Labs. It is for recruiting the best of the best is what you want. But Dave and I were both very NIH oriented, and we basically set out a plan it was a 5-year plan to acquire MSTP status for our MDPhD program. And what that is basically is acquiring a training grant that is reviewed by other MDPhD directors. And basically what you're showing them is that you have a very integrative program, very good students, very productive students. And the way the story went is that in 2004, Dave became the chairman of anesthesiology. And about a year later, he realized that, of course, anesthesiology is a huge clinical department, over 100 faculty, that it was just a little bit too much for him to do both the chair of anesthesiology and the director of the MSTP program. Is that he at that time asked Mike Dunn to transition us, where I would become the director of the MD-PhD program and he would be the associate director. So we essentially switched positions. And I was a little bit leery of that because it was I was a PhD directing an MD-PhD program, and I had to do some homework. And as I did my homework, I found that there are about 100 MD-PhD programs in the country, and about a third of them are directed by MD-PhDs, and a third are directed by MDs, and there are a third of them directed by PhDs. So when Mike Dunn asked me to be the director, that was my question, and I didn't actually say yes right away until I knew that it was appropriate to be the director. But I was asked, and I did accept, and that was a big it was a big let's see, I would say commitment on my part, because up until that time I was pretty much a researcher, and I hadn't had a lot of administrative training. And when I took the program over, we had about, Carol Knapp, who's our administrator, she says we had about 22 students in the program. And so I was now responsible for 22 students that were clearly brighter than I was. And the only thing I had over them was a little bit of experience, I think. But it was a great time because we maintained the focus of the program towards obtaining this training grant. And one of the things that I did, not being a physician and running this program, is that I got involved in the AAMC. That's the Association of American Medical Colleges, and I think I was just the right person at the right time, in that I was able to get involved in a committee that one of the that was part of the MD-PhD Association. It was called the Communications Committee, and maybe I've been involved in that committee for about 10 years, and of those 10 years, I've been the chair of that committee for about 5 of those years. So it's given me some visibility in the MD-PhD community. And we basically go out and we give seminars and workshops for students that are thinking about MD-PhD as a career. So it was really nice, and it got me involved in the AAMC, which is the part of a medical school that a PhD rarely interacts with. And I eventually became, I was the chair of the MD-PhD section at the AAMC for maybe about 5 years ago or so. And that was a real honor to be recognized by my colleagues to take that position, because it's typically a position that is an MD-PhD type person. So I was really honored to do that. But and it gave me a lot of exposure, a lot of best practice, how to train MD-PhD students, and we were really fortunate. In 2010, we received our first training grant, our T32 for the MSTP, and just last year we had our first renewal. So we're in our second round of training for that, and right now we've been able to grow the program to the point where we have 50 students in training, and it's I do that with Carol Knapp, who's our administrator, and Carol has been our administrator since the year 2000, and I have 3 really great associate directors who give us a medical link, and that's Gil White, who's also the president of the BRI. He's an MD, card-carrying MD, and Cal Williams, who's a researcher in pediatrics and the research director of the CRI, Anita Salzman, who's an MD-PhD also, who's a gastroenterologist. So that's the group that we use to train these MD-PhD students, and I think we have a really nationally recognized medical scientist training program at MCW now, and it's something that I'm really proud of the students, and I'm proud of the administrators, and I think I'm really fortunate that there's so much support for the program in both the basic science departments and the clinical departments. So that's been a real honor for me to be the director of that program for the last couple of years. So it's a nice time.
Now, we talked before about Dr. Grossberg had been the chair of microbiology, and then when he stepped down and retired, Paula Trackman came in.
Yep.
How did the department change? I know the name of the department changed.
That's right.
And did the emphasis within the department change? Were there new recruits as well?
Yeah.
And
yeah, Paula was really, I'd say she was just the perfect person, both personality-wise and scientifically, to join MCW. She came from Cornell Rockefeller. She was I think she had just transitioned to a professor's position at Cornell, and we were really fortunate to have her come into the department because she really was a driver, and she was very vocal for the department. I think she recruited really well. We have some really exceptional faculty who are at the professor level, associate and assistant professor level. So I think Paula was a really good recruiter. I think she thought really well. She was able to maintain her research program while she was the chair. What I like to say is that in the last 50 years, our microbiology department has had 2 chair people, Sid Grossberg for 33 years and Paula Trackman for for 17 years. So I think it's a very unusual situation, but I think they gave us a really good stability for faculty development over those years. So I've been the interim chair since Paula left, and I've been able to I've had the opportunity to help the faculty continue to develop, and I'm really impressed with the quality of the faculty that we have in the department. So so we're really excited about actually the next era of the department, and I'm really looking forward to bringing having a permanent chair that'll help build the next component. Microbiology continues to be just an exceptional field to study. The microbiome is developing. It's all of the microorganisms from those in your mouth, to those that live in your stomach, to those that live in your gut, to those that live on your skin. And what we're realizing now is that they're not just commensal relationships where they live and we live. They really influence a lot of our metabolism and our health. So we see the microbiome as being the next brain initiative at the NIH.
And it's also a scary time too,
Yeah.
as we see the growth of the superbugs that
That's
we're
right.
dealing with, and you'll be on the front line in terms of trying to find the answers for those.
And I think we're very fortunate in our department. We have Chris Christich, who studies antibiotic resistance in the enterococcus, which is a very important hospital-acquired infection. And he's been able to sort of focus that in a collaboration with Nita Salzman in looking at the effects of the enterococcus in the microbiome. So I think we have some really exceptional investigators that are really going to drive research at MCW, and it's fun to watch them work together. We have a very nice group of herpes virologists that have some really nice collaborations together. Vera Tarakanova and Scott Terhune, to name a few, but it's a really nice collaborative group, and Rich Robinson and Tom Zart study Mycobacteria tuberculosis as a microbial pathogen. So I think the scope of our research is really it continues to grow, and we're really looking forward to the next iteration of microbiology. The other thing that we're trying to do now is to emphasize immunology in microbiology, because what we often forget is that essentially all of the immunologists at MCW are secondary faculty in our microbiology department. So the graduate program is actually microbiology, immunology, and molecular genetics, though the department is microbiology and molecular genetics. And we changed that because the interest in immunology is almost exponential in graduate training. And we've been very fortunate to have a very strong immunology group, and they interact with us at the departmental level also. So all of the graduate students that train in immunology actually train in our department. So we don't think of immunology as being part of the department, but it's actually a very important component because immunology has a tremendous influence in not only basic research, but also clinical and translational research.
Well, now, as MCW has started to expand, we have new campuses in Green Bay and in Wausau. We've created a pharmacy school now. We're looking at becoming a health sciences university. Are there opportunities for other joint degree programs within MCW as well? Beyond the MD-PhD, is there a possibility that we may have a PharmD program married with a PhD or with an MD? Is there a MPH program that could, what are the opportunities?
So the activities that and I'm very interested in that because dual degree is something that I think about a lot. I have spoken to the pharm leadership about the possibility of doing a pharm PhD. I think that's something that we'll see evolve for a smaller subset of the pharmacy students, and I think that's a great opportunity to do biomedical research as with that dual degree. The other area of dual degree that I think we'll see expand at MCW is the MD-MS, which is a medical degree with a master's in science, and that's a spin-off of the clinical translational program that we have, our CTSI. In the last renewal of the CTSI, we were able to secure a TL1 training grant, which is specifically to support students that are either going to complete a PhD or to complete a master's. And so the individuals that are supported off of that training grant are PhDs and MDPhDs who are completing a PhD, but also MD residents who are completing a master's and medical students who will complete a master's during their training as a medical student. So we see this as being a really important part of the biomedical workforce, the MD-MS, especially in the clinical translational arena, as part of the concept of team science. We have a PhD, an MD, an MD master's, and a resident, and a medical student as part of a research team. So we're really excited about some of the dual degree programs that are based off of biomedical research, not just a PhD, but also the master's.
You came here in 1984 when this was a small institution. Mm-hmm. Research was really just getting off the ground. Mm-hmm. Everyone knew each other. It was an institution that really had a limited identity. We weren't really on the map in terms of mm-hmm. where other researchers at other institutions knew who we
right.
were.
Numerically. Right.
And now
right.
today, we are a significant research institution. We are known in the scientific community for the work that we're doing very
right.
well. When you look back on the years here, what's the same and what's different? Yeah. How has the culture changed here, and what is inherent in the culture today that existed here in 1984?
Yeah, I think there are two things that remain the same. And the two things that remain the same are the collaborative nature of the faculty, which is really pretty remarkable, considering the size of the institution now. We really seem to attract individuals that are very collaborative in their nature. And the other, which is really important, is that we have clinical chairs that are really supportive of biomedical research, and to the extent that they support biomedical research. So it isn't just saying that they are interested in it. They are interested in recruiting faculty in a clinical department that's going to be a biomedical scientist. They're interested in supporting biomedical research. So we've been really, and when I talk to my colleagues outside of MCW, the biggest concern they have is the lack of support they get from the hospitals that are associated with their academic medical centers. And I think that's one of the things that really makes MCW such a great institution, is that we've had a long history of chairs of anesthesiology and medicine and pediatrics and the Eye Institute that have supported biomedical research over the 30 years that I've been at MCW. Rob Lane and the chair of pediatrics, and Roy Silverstein, the chair of medicine, and of course Dave Waltier in anesthesiology, all of them very supportive of biomedical research, and the new chairs that we recruit also very supportive. So that's remained the same. I think the things that have changed, when I came, we didn't have any centers. Now we have some really spectacular research centers integrating research and clinical sciences. I think that's really helped MCW. I think that's linked some of the clinical departments with the basic research departments. I think a really good example of that is what goes on at the Eye Institute. Dale Hoyer, who's the chair of the Eye Institute, is really he's a phenomenal clinician, but he just has a passion for research, and he has really built a very productive research component to the Eye Institute. So and that's been really consistent for the last 20 years. So we've been really, I think, really very fortunate in how much support we've had from the clinicians, and in the light of NIH funding being, as I started, being very lucrative, a lot of support for biomedical research to the 90s, where it was actually very difficult to get supported, to the 2000s, where it began to be supported again, to now where it's really a challenge to maintain a research effort at an academic medical center. I see MCW as really being poised to continue that upward trajectory, just based upon the leadership and the faculty that are here and the faculty that we've recruited. So I'm really optimistic with MCW, where we started and where we are now, and as we move into the future. I think there aren't very many freestanding academic medical centers, but if you look in today's environment, I think it really gives us the unique ability to respond to changes in the environment. And again, that's all based upon the congeniality between the clinical departments and the basic science departments. I think it's really a special place. I mean, I've been here for 30 years. I've been really fortunate in the support I've had, and the great students and great collaborators, and I see it as a very bright future. What I've really enjoyed is having the ability to identify great colleagues nationally and internationally, but also having a core of colleagues at MCW who are part of a team. And I think our microbiology and genetic and molecular genetics program is spectacular, and it's been just a great place to train. And I think I've really been fortunate in that I've had the opportunity to be involved in the development of a number of these training programs. So
Great.
yeah.
Thank you.
Okay.
Appreciate it.